The university shutdown over the summer months was really frustrating, as I was keen to get valuable research experience to help decide my future career direction. Luckily, I spotted the BSPP lockdown bursary advertised on Twitter. I was able to contact one of my lecturers who I knew had an appropriate project using RNA sequencing to identify changes in gene expression and small RNA regulation as a factor of temperature that I could work on to develop my computational biology experience. As a biological scientist student, I was excited to study the interaction between plants and pathogens in the Rhizosphere, as it brought together a range of material from across my course. I've been working as part of a large project looking at the effect of temperature on tomato root exudates and the interaction with microbes and parasites in the soil. We know that temperature alters root exudate composition and Rhizosphere interactions, but we don't yet know how changes in temperature are relayed through changes in gene expression. I've been learning how to code in R and use a variety of bioinformatics software packages to analyze RNA sequencing data and small RNAs using the high-performance cluster at QUB. I've analysed millions of sequencing reads, and while this can be daunting at times, I've enjoyed overcoming the many setbacks and frustrations. I've learned that biological data can be surprisingly variable, even with high levels of replication. I have identified new non-coding RNAs in tomato, and degradome analysis has revealed a significant role for transacting small interfering RNAs in the regulation of gene expression as a factor of temperature. Looking at patterns in the data and exploring published papers to come up with new ideas is both equally challenging and enjoyable. To be able to work as part of a research team, producing information for others to work on requires an attention to detail and the elimination of mistakes that can be laborious, but also satisfying. The opportunity provided to me by the BSPP and the Dalzell laboratory at Queen's University Belfast has reinforced my desire to continue in the field of bioinformatics. I now have a more specific understanding of the skills I will need to develop during my last years at university. I am more aware of my need to gain experience in experimental design, statistics, and at the laboratory bench. This will allow me to more fully understand the process of experimentation. Yes, this year I was particularly interested to learn about the work my supervisor carried out in Africa, funded by the Gates Foundation, on the huge problems caused by plant parasitic nematodes. This project has shown me that the study of fundamental molecular biological questions can have real consequences for the world around me, offer new opportunities to show how genetic engineering could be used in the fight against plant pathogens.